Before we go anywhere, you need to know that you don't actually have one type of sweat gland. You have two, and they behave completely differently. The first type is called Eccrine glands, and you've got roughly 2 to 4 million of them spread across nearly your entire body, and their job is simple: temperature control. They produce a thin, watery fluid that's about 99% water, with trace salt and minerals, and it is, chemically, essentially odorless. This is the sweat cooling you down on a run. The second type is apocrine glands, and these are concentrated almost entirely in specific zones: your armpits, your groin, around your nipples, and they don't activate for temperature at all. They activate for stress, emotion, and puberty, and they release a thicker, protein and lipid-rich fluid, still odorless on its own. The difference is that apocrine sweat is basically a buffet, and the moment it hits your skin, resident bacteria go to work breaking down those proteins and lipids into smaller compounds, and those breakdown compounds are what you actually smell. You've spent your whole life thinking you smell like sweat. You've actually been smelling bacterial waste products the entire time. Foot odor is caused overwhelmingly by a specific bacterium called Brevibacterium, and it produces a compound called isovaleric acid when it breaks down sweat and dead skin trapped inside a shoe. Here's the thing nobody tells you: Brevibacterium lenins is the exact same bacterial strain, or one nearly identical to it, that's deliberately used to wash and ripen certain soft cheeses, including Limburger and several classic French washed rind cheeses, to give them that same pungent, sock-like smell on purpose. A widely cited 2006 study out of NIZO Food Research in the Netherlands, led by food microbiologist Joris Winkel, confirmed that mosquitoes are actually more attracted to Limburger cheese than to a resting human foot, and used that finding to help design better malaria trap technology in parts of sub-Saharan Africa, because Anopheles gambiae, the primary mosquito species responsible for malaria transmission, tracks human targets, using foot odor as one of its main homing signals. Sound familiar? That single, seemingly gross fact about your socks led directly to public health mosquito traps deployed in real villages, baited with cheese instead of human bait, to pull disease-carrying mosquitoes away from actual people. Bad breath, medically called halitosis, works on almost the exact same principle as foot odor, just relocated to your mouth. The primary culprits are volatile sulfur compounds, mainly hydrogen sulfide and methylmercaptan, produced by anaerobic bacteria living on the back of your tongue and in the small gaps between your teeth and gums, feeding on trapped food particles and dead cells. An Israeli dentist and researcher named Mel Rosenberg, working out of Tel Aviv University starting in the late 1980s, essentially built the entire modern scientific field of halitosis research from almost nothing, developing the first reliable gas chromatograph-based measurement tools to actually quantify bad breath instead of relying on someone leaning in and guessing. Rosenberg's research found that roughly 80 to 90% of chronic bad breath cases originate specifically from the tongue, not the teeth, which is exactly why tongue scraping, something Ayurvedic practitioners in India have documented in medical texts for over 3,000 years, turns out to be more effective at reducing halitosis than mouthwash alone in multiple modern clinical trials. Nobody tells you about garlic breath specifically. Brushing your teeth barely touches it, and there's a very specific chemical reason why. When you eat garlic or onions, your body absorbs sulfur compounds like allyl-methyl sulfide directly into your bloodstream through digestion, and your body can't fully metabolize it quickly. Instead, it gets carried through your blood to your lungs, where it's exhaled with every breath, and it also gets excreted through your skin's own pores as you sweat, which is why heavy garlic eaters can smell garlicky from their skin up to 24 to 48 hours after the meal, according to research out of Ohio State University published in the early 2010s tracking allyl-methyl sulfide levels in breath samples over time. You're not smelling leftover food in your mouth. You're smelling your own bloodstream, ventilating garlic out through your lungs like an exhaust pipe, which is exactly why mints and toothpaste barely make a dent. The source isn't your mouth, it's your entire circulatory system. There's a rare inherited condition called Trimethylaminuria, sometimes nicknamed fish odor syndrome, caused by mutations in a gene called FMO3, which normally produces an enzyme responsible for breaking down a compound called trimethylamine, a byproduct of digesting foods like eggs, certain legumes, and saltwater fish. In people with a functioning FMO3 gene, trimethylamine gets neutralized in the liver before it ever reaches your skin or breath. In people with a mutated version, it doesn't get broken down at all, and it builds up and gets released through sweat, urine, and breath, producing a persistent, unmistakable smell of rotting fish that has nothing to do with hygiene and everything to do with a single malfunctioning gene. The condition was first clinically documented and named in a 1970 paper by British biochemist R. L. Humbert, and it's now estimated to affect roughly 1 in every 10,000 to 40,000 people globally, though researchers believe it's significantly underdiagnosed because patients are often mistakenly told for years that the smell is simply a hygiene problem, leading to real documented cases of social isolation and even job loss before someone finally traced it back to a genetic test. Here's one that used to function as an actual diagnostic tool long before modern lab equipment existed. When someone's body is in a dangerous metabolic state called diabetic ketoacidosis, usually an undiagnosed or poorly managed type 1 diabetes, their body starts breaking down fat for energy instead of glucose, producing chemical byproducts called ketones, and one specific ketone, acetone, is volatile enough to be exhaled directly through the lungs, producing a breath odor doctors have described for well over a century as sweet, fruity, or like nail polish remover, because acetone is chemically the exact same compound used in nail polish remover. Physicians were documenting and diagnosing this specific breath smell as a marker of severe diabetic crisis as far back as the late 1800s, decades before blood glucose meters existed, meaning a trained doctor's nose was, quite literally, one of the earliest functioning diagnostic instruments for a life-threatening endocrine emergency. You've got mosquitoes tracking foot bacteria to find prey. You've got doctors tracking acetone on someone's breath to identify a medical emergency. Different smells, completely different stakes, same underlying idea, odor as information. And here's the twist almost nobody knows about. A meaningful percentage of the human population produces essentially no underarm body odor whatsoever, and it comes down to a single gene. In 2006, a Japanese research team led by geneticist Kichiro Yoshiura at Nagasaki University identified a specific variant of a gene called ABCC11 that controls whether apocrine sweat glands secrete the fatty precursor compounds that bacteria need to produce odor in the first place. People carrying the inactive version of this gene produce dry, flaky earwax instead of the wet, sticky kind, and their apocrine sweat is essentially odorless because there's nothing there for bacteria to break down. Yoshiura's study found this inactive variant is present in roughly 95% of East Asian populations, but only around 0.6 to 3% of populations of European and African descent, making body odor, in a very literal genetic sense, unevenly distributed across humanity, which is also, incidentally, part of why underarm deodorant marketing and usage rates historically differ so dramatically by region, something anthropologists studying consumer behavior have written about directly citing Yoshiura's genetic findings. And then, there's the category that reframes everything you've just heard: body odor as an unconscious signal in mate selection. In 1995, Swiss zoologist Klaus Wedekind ran what's now famously known as the sweaty t-shirt study at the University of Bern, having male participants wear the same cotton t-shirt for 2 consecutive nights without deodorant, cologne, or scented soap, then having female participants smell the shirts and rate them by pleasantness. Eccrine found that women consistently rated the body odor of men most pleasant when those men had genetic immune system markers, specifically in a gene complex called the major histocompatibility complex, or MHC, that were most different from their own. A pattern researchers believe evolved because offspring from genetically diverse immune profiles tend to have stronger, more varied immune resistance. The one exception researchers found was striking. Women taking oral contraceptives at the time of the study showed the opposite preference, favoring men with more similar MHC profiles to their own, a finding that's been referenced in dozens of follow-up studies about hormonal birth control's potential influence on partner selection ever since. You've spent this entire time thinking of body odor as something to eliminate. Evolution has been quietly using it as a communication channel the whole time.